Everybody, welcome. So I'm Abby from Precisionary Instruments, and I am honored to welcome Dr. CynthiaKoziol-White today, who is our webinar speaker. So a little bit about Professor Koziol-White. She received her graduate degree from the University of Wisconsin-Madison in Cellular and Molecular Pharmacology, where she examined the mechanism of eosinophil survival and activation in allergic asthma. She then moved from the Midwest here to the East Coast, where her first postdoc fellowship was with Dr. Angela Haczku at University of Pennsylvania. There, she studied the ozone and allergen-induced mechanisms of dendritic cell activation in the lung. Her second postdoc was with Dr. Reynold Panettieri Jr. at University of Pennsylvania, where she had been examining airway physiology and modulation of airway smooth muscle function in asthma and other respiratory disorders. So Dr. Koziol-White has a huge repertoire of publications, including 59 peer-reviewed journal articles and 5 book chapters. And what is really impressive is outside of just writing and research, she also has served as a mentor to about 35 students at various stages of their career. So we're really lucky here to have her at our webinar, where she is going to discuss the versatility of the precision cut lung slice, or PCLS, a system that she has actually helped develop and utilize to study airway function for almost 20 years. So with that, Dr. Koziol-White, I want to hand it over to you, and thank you so much for being here.
Thank you so much for the invitation to speak, and for people coming to the webinar. Can I have you enable screen sharing so that I can get my presentation up?
Absolutely. And for everybody else, please do feel free to add comments, questions, anything else in the chat box. I'll be monitoring that. And once we are done with the presentation, we're going to either have you unmute yourself to ask questions, or I can read off and ask the questions for you. And Dr. Koziol-White, we're seeing the presenter view.
Okay, so is that better?
Still presenter view. Now it's perfect.
Oh, okay, cool. All right, so so what I'm going to talk about is using lung slices as an ex vivo model to study lung diseases. I'm not going to cover anything that has to do with lung cancer, but more functional things that we study in terms of obstructive lung diseases. So we and others have used PCLS as a model to study peripheral airways and study ex vivo effects of exposure in an integrated system. So what's great about this system is that you not only have the structural cells of the airways, but you also have some lung resident immunocytes and things like that. And this system actually maintains an in vivo mechanical environment to study contractility, relaxation, and also inflammatory mediator release, which is the main sort of things that our lab studies. We found that it's advantageous over using airway smooth muscle strips or single cells in order to understand sort of the integrated tissue response, and sort of with the airway smooth muscle sort of in its native environment. It's been used for not just human lung slices, but also rodent lung slices. There are many publications on murine lung slices and rat lung slices, and it's been used to study the impact of a number of environmental triggers like allergens, respiratory viruses, intoxicants, and things like that, on inflammation, a remodeling of the airways, and then also contractility and relaxation. So the way that we generate these lung slices in our lab is we take otherwise healthy human lungs. So they are from donors that have no history of chronic disease, definitely no history of chronic lung disease. We also actually use lungs from other disease states. People that have chronic obstructive pulmonary disease, severe asthma, people that actually have died of their disease, and then things like idiopathic pulmonary fibrosis and sarcoidosis, which is also another fibrotic lung disease. We dissect off the lobe and then inflate with a low melting temperature agarose. We actually take it and then section it and cut it perpendicular to the way that the airway is running from sort of the larger bronchi. We take a cork boring tool essentially and visually determine where there's a small airway that's about 1 to 3 millimeters in diameter. The cork boring tool has a diameter of about 8 millimeters or so. We take these cores of tissue, feed them into the Precisionary instrument's vibratome, and then slice to about 350 microns. It's sort of variable depending on which group you talk to, what they will slice to. We found that 350 microns is probably about as good as we're going to get in order to get penetration of the tissue with things like small molecule inhibitors, G protein coupled receptor agonists and things like that. Whereas some of the thicker slices and stuff that other people make, you may not necessarily have as much penetration into the tissue as you would like, but it actually is able to keep its structural integrity at 350 microns. And then what you see the bottom left two pictures is the lung slice with a platinum weight that has nylon attachments. And it's not necessarily to keep tension on the tissue like there are in like organ baths to measure contraction of muscle, but it's more to make it so that it kind of stays in place and it doesn't just float away. Because the parenchymal tethering surrounding the airway of the alveoli tissue actually kind of keeps the airway in sort of almost like an L-knot type of state, which you would have in a tissue bath, and you can locate the small airway on the slice. In order to measure contraction and relaxation of the airways, what we do is we take pictures, and we take pictures every, in this case with carbachol, which is basically like an acetylcholine derivative. We take pictures every about 10 minutes, which we found 10 minutes is kind of a plateau in human lung slices to measure the contraction of the airway. So we measure that luminal area over a dose response of a contractile agonist. And as you can see, as it narrows, it increases the amount of contraction. So it's expressed as a percentage of baseline. And then on the sort of flip side of that, we can also use asthma therapeutics like bronchodilators or beta-2 receptor agonists, like isoproterenol is one of those. And you can actually see as you increase the dose of isoproterenol, you actually increase the luminal area. So then the amount of contraction or whatever of the airways actually decreases. And we've expressed this as a decrease in the amount of contraction, or on the flip side of that, like an increase in the amount of bronchodilation or relaxation of the airways. We've used this to study anything from just basic mechanisms of airway contractility and relaxation. There was a graduate student that we had in the lab, I believe it was maybe 2014 is when he had started. And he was interested in trying to understand mechanisms downstream of muscarinic receptors, which respond to carbachol or acetylcholine derivatives in terms of the signaling pathways that are activated. And what he had found is that G-alpha-12, a small G protein, actually will induce activation of Rho kinase or Rho, and then also Rho kinase to essentially inhibit a phosphatase that would normally dephosphorylate myosin light chain. And this basically sustains contraction. So there's sort of two different pathways to induce contraction or keep contraction going. And he found one out of the two in airway smooth muscle. And so what we did with the lung slices is we actually used a Rho kinase inhibitor called Rosin, and showed that it's roughly equivalent to what you would get in terms of relaxation response compared to formoterol, which is an industry standard long-acting beta agonist. So the formoterol is the one that's actually used in treatment of asthma. We extended these sort of novel mechanisms of airway contraction and relaxation by using these Bay compounds or Bayer compounds. What they are is they're actually soluble guanylyl cyclase activators. The traditional way for you to dilate the airways is by using a beta agonist, which is used in asthma and COPD treatment. In the diagram to the right, you'll see that formoterol activates the beta-2 receptor and activates soluble adenylyl cyclase, which then goes on to produce cyclic AMP and then induces relaxation of the airways. And that's sort of the canonical bronchodilation pathway. We wanted to evaluate whether production of cyclic GMP, which is another cyclic nucleotide, through activation of a soluble guanylyl cyclase using these Bayer compounds would also induce dilation. And what we found is that in comparison to a beta agonist formoterol, you actually get pretty much equivalent amount of bronchodilation or relaxation of the airways if you use something that activates soluble guanylyl cyclase. To extend sort of our, our studies of novel mechanisms of contraction or relaxation, we also wanted to assess whether or not activation of signaling mechanisms that are downstream of contractile receptors or contractile agonist receptors, if you inhibit them, do you actually see a relaxation of the airway, or does it take more than that in order to elicit dilation? So what we found is that using the P110 PI3 kinase inhibitor, it's a broad spectrum inhibitor of PI3 kinase, the LY294002, and then also a delta P110 selective inhibitor called KEL-101, that you actually see you're able to induce relaxation or dilation of the airways, pretty much equivalent to formoterol. And then again, another inhibitor downstream of PI3 kinase, where you have Rho activation following contractile agonist stimulation or carbachol stimulation. If you use Rho kinase inhibitor 27632, you actually again get dilation of the airways. So this shows you that by probing something downstream of a contractile receptor, you can actually, and using an antagonist of signaling molecule, that you can use small molecule inhibitors to actually induce some similar effects to what a beta agonist actually induces. So we thought that was kind of a cool concept. And then there's sort of an interesting and novel bronchodilators study. So it was found by some of our collaborators that G protein coupled receptors called TAS2R, or bitter taste receptors, are present on airway smooth muscle. And then what these investigators found is that it actually, agonism of the receptors, bitter taste receptors, actually induces relaxation of airway smooth muscle, and it's irrespective of generation of cyclic AMP, which is kind of a little like counterintuitive, I guess, in the field, that it would be a calcium dependent mechanism or something like that, that these receptors act through. But needless to say, activation of these receptors in the lung slices actually does induce relaxation, as shown on the right. So quinine is actually a bitter taste receptor agonist. And in the presence of IL-13, which is an allergic a mediator that's produced in allergic asthma, you still get relaxation of the airways. However, if you use a beta agonist, you actually see an attenuated relaxation. So this would suggest that these types of agonists of this receptor might be a really great way to induce relaxation of the airways that may not necessarily be modulated by the inflammatory environment that's there. So that's kind of I guess, a cool and novel pathway. And more recently, we've shown that changes in metabolism actually have an effect on contractility of the airways. So this PFK15 is actually an inhibitor of glycolysis. And what you can see down on the bottom right is that you can measure glycolysis actually in the lung slices themselves. And as you increase the amount of the PFK15, you inhibit almost down to nothing in terms of the amount of glycolysis that occurs. Interestingly, if you take it and you use the lung slices and pre-incubate with the PFK15, you actually see what we call bronchoprotection. So what it does is it basically sort of inhibits the ability of the airways to contract. And it's pretty much on par with formoterol response. So it's been established for quite some time that like sort of a pre-incubation with formoterol or like a continual administration of formoterol actually decreases the sensitivity of the airways to contraction. So like it makes them less twitchy. So by inhibiting a metabolic process with PFK15, you actually get a very similar response, which is kind of a novel finding. We also have used lung slices to study the effects of different therapeutics that are used in asthma. So it was clinically, it's been established for quite some time that overuse of bronchodilators, so like your rescue inhalers and stuff like that, actually causes downregulation of the beta-2 receptor that would normally respond to the therapeutic. So that's why if somebody is taking a number of different puffs of a rescue inhaler and they're not getting any relief from it, they have to go to the hospital or else their airways will stay closed. So this is sort of an illustration of molecular mechanisms and how we can use this ex vivo system to actually study this effect. So what you see in the plot on the left-hand side is that in the triangles, albuterol is a short-acting beta agonist. So that's what's a near inhaler. If you give that for a minimum of 6 hours to the lung slices prior to contracting and then relaxing the airways to another beta agonist, in this case, it's isoproterenol, you actually see an attenuation of the ability of that beta agonist to induce relaxation of the airways. However, if you pre-incubate with dexamethasone, which is, it's not as commonly used clinically so much now, but other steroids that are more selective have taken its place. It's an example of a glucocorticoid. So if you pre-incubate with dexamethasone prior to giving the 6 to 24-hour albuterol treatment, you actually rescue the effect. And what was interesting is that we had then used a YFP labeled lentiviral vector for the beta-2 receptor, and showed that if you look at the pictures on the right-hand side of the screen, you actually see once you give a beta agonist treatment, there's a lot of formation of punctate foci in like the cytoplasm of the smooth muscle. So like the long sort of stringy looking thing is actually a smooth muscle that's in one of the lung slices. So you see that there's an internalization of the receptor that we think, and or others have shown that that it's desensitization of the receptor, so it's not able to actually respond to this therapeutic anymore. And then interestingly, it's a therapy that's been more closely studied and more widely applied in Europe, and it's starting to kind of gain traction in the United States. But there's this thing called SMART therapy, and it's a combination inhaler that has a long-acting beta agonist and also has a glucocorticoid in it. And what they've been using it for mainly in the United States and then also in Europe is for control of asthma symptoms. So these would be taken once a day or something like that, maybe twice a day, in order to keep the sort of twitchiness of the airways at a minimum. But the SMART therapy actually uses it not just as a controller therapy, but also uses it as an acute therapy. So like uses it as a rescue inhaler. And what's been shown clinically is that the combination of the steroid plus the beta agonist used acutely and also as control, there's a greater amount of relaxation of the airways and greater efficacy of like the therapeutic in general. And we've used the lung slice system to basically mimic this. So what you see on the left and then also on the right is that if you combine budesonide with, which is a glucocorticoid, with formoterol, which is a beta agonist, you actually get a greater amount of bronchodilation or relaxation of the airways compared to just the beta agonist itself. So this is sort of a proof of principle thing. We've also used lung slices in the study of asthma, so Th2 mediated disease. So Th2 is sort of the inflammatory environment that's induced in allergic asthma. So there's a number of different inflammatory mediators that are released into the lungs of allergic asthmatics. Among them are TNF-alpha and IL-13 or interleukin 13. And you actually see a bit of an increase in the maximal amount of contraction that's induced if you give TNF-alpha and then use a bronchoconstrictor. However, you see a really robust increase in the amount of maximal contraction that's induced by carbachol if you have IL-13 present. And with both TNF and IL-13, you actually shift the curves to the left, which means that you're increasing the sensitivity of the airways to a contractile agonist. So you're basically making the airways twitchier. And then, as I alluded to before when I was talking about the quinine results, if you stimulate with IL-13 and then you look at relaxation of the airways to a bronchodilator, you actually see an attenuation, which is not there if you are stimulating with a bitter taste receptor agonist. We've also used lung slices studying mechanisms of allergic asthma. So what's really interesting and I think is kind of cool is that, like I had mentioned, I think at the beginning of the talk, that there's all sorts of different cell types that are present in the lung slices themselves, whether it's in the airways or it's the surrounding parenchymal tissue. And so what we've done is we've taken advantage of that sort of thing. And it's about maybe about 30% to like about a third of the population or whatever is allergic. So we sort of take advantage of that in the sense that we are able to stimulate these things called mast cells that are responsible for allergic, basically a response to allergen. The way they respond to allergen is that there's a ton of immunoglobulin E that is produced. And there's receptors on the surface of mast cells called the Fc epsilon receptor, which is a high affinity IgE receptor that will recognize this. And in the normal allergic asthma, you would have IgE that's specific to a certain allergen, whatever the person's allergic to. However, we don't know what the person that we get the lungs from is allergic to. So we sort of fake the response, if you want to put it that way. We take an antibody that will recognize the IgE, so it's an anti-IgE, and it basically does the same thing that the allergen does in the context of an allergic asthmatic, like reaction of the lungs. So what it does is it actually clusters the Fc epsilon receptor and causes activation of mast cells, and then that induces things like degranulation. So you have release of lipid mediators, histamine, inflammatory cytokines, all sorts of different things that basically either induce bronchospasm itself or causes recruitment of different immune cells to the airways and things like that. So what you see on the top left is that you see that if you give an IgE overnight and then cross-link with an anti-IgE, you get about maybe 50% constriction compared to like the baseline airway measurement. If you use dexamethasone, which I said is a glucocorticoid, it's a steroid, you find that there's absolutely no difference, which is actually consistent with a lot of the literature and stuff, that steroids really just don't have an effect on mast cell mediated degranulation and things of that sort. However, if you take fexofenadine, which is a histamine 1 receptor antagonist, or you use montelukast, which is a cysteinoleucotriene receptor antagonist. So it's a lipid mediator receptor antagonist that they're lipids that are derived from arachidonic acid. If you use either of those two inhibitors, you actually inhibit partially the response that you see if you cross-link and induce constriction that way. However, if you use an inhibitor of Syk kinase, which is a kinase that is downstream of the Fc epsilon receptor, but is very membrane proximal, it's like one of the first events that occurs following activation of that receptor, you pretty much ablate the response that you see. Interestingly, it had been shown previously by other groups that if you use serum from allergic patients and you basically do the same sort of sensitization procedure that we do with the IgE. And then these people use the sensitization with the serum, and then they came back with the allergen that that person was allergic to, and they actually saw constriction of the airways, whereas we use a general, anti-IgE in this case. We've also utilized a ligand for an inhibitory receptor. So CD33 is an ITIM receptor. It's an inhibitory receptor, and we found that if you cross-link the Fc epsilon receptor, in this case, we actually used an IgE that was specific to a thing called TNP. If you incubate or pre-incubate with this CD33 ligand and TNP, you actually get inhibition of the response, which we thought was kind of a cool thing, because it's showing that you do get immune cell activation in lung slices, and you're actually able to modulate constriction through this pathway. Other groups have used lung slices to study COPD. So there's a group out of the Netherlands that had showed that lung slices that are derived from COPD patients compared to control, like non-disease patients, actually show an increase in contractility of the airways. And what they attributed it to was not necessarily because there was more smooth muscle in the airways, but it was because the parenchymal tethering of the surrounding tissue wasn't there to sort of hold it in place. So you got an increased contraction just because there's just not a retractive force on the airways. So it's another great area of study. We've used lung slices to study exacerbations of asthma and COPD. So in this case, we used a viral mimic of TLR3 agonist called poly-IC that's pretty ubiquitously used in terms of like respiratory virus exposure as a mimic. What we found is that it was able to induce a whole host of inflammatory mediators that were released from the lung slices. However, it had little to no effect on modulating contraction or relaxation of the airways. So we feel like that it's not necessarily the best model to actually study the effects of virus exposure. However, future studies, we actually used intact rhinovirus. So we used an RVA39 strain. RVA and C serotypes are the ones that are most commonly associated with severe exacerbations or worsening of symptoms of asthma and COPD, and found that in exposure of non-diseased tissue or non-diseased lung slices compared to ones that were derived from mild to moderate asthmatics, that it basically didn't change the contraction of the non-diseased tissue, but in the asthmatic tissue, it actually increases sensitivity to a contractile agonist. We also used RVC serotypes. So in this case, we used RVC15 and found that actually in the normal tissue, the non-diseased tissue, there was an increase in the maximal amount of the maximal effect, as well as an increase in sensitivity of the airways to a contractile agonist. And then more recently, and this is hopefully going to get published soon, it's in revision, we actually used it and used RVC15 and then evaluated relaxation to a beta agonist and found that there was an attenuation of relaxation following RVC15 stimulation. We and other groups have used lung slices to study another type of exacerbation. So in this case, things like gaseous environmental contaminants or environmental toxicants. So we had used the lung slice system. We had used not human lung slices, but mice. We had used either exposure of mice, an acute exposure to ozone, or actually ozone exposure of the lung slices themselves. Showed an increase in airway hyperactivity, so an increased responsiveness to carbachol stimulation that we found was cytochrome P450, but not cyclooxygenase 2 dependent. It's been shown by others that ozone and nitric oxide were able to induce inflammatory mediator release from murine lung slices. But for the most part, there hasn't been a heck of a lot of research that's been done to examine ozone or environmental gases in terms of changes in contractility or inflammation in human lung slices. There was a publication that came out of the Panettieri group some years ago that showed that formaldehyde exposure, so things like you would encounter formaldehyde in things like actually shampoos, and then also building materials have some formaldehyde in them, so that you can get environmental release, like depending on the building that you're in. But we found that formaldehyde exposure actually induced an increase in responsiveness of the airways to a contractile agonist, and found that it was Rho kinase dependent. And then there was a study that was done by a German group that looked at industrial toxicant exposure. And what they found is exposure to this thing called ammonium hexachloropalatinate, which is, again, an industrial, it's released as a result of some industrial processes, actually induced release of a number of different inflammatory mediators, including TNF-alpha, IL-13, eotaxin-2, and IL-5. And all of these mediators that are highlighted are actually mediators that are involved with allergic asthma. So they're Th2 and like inflammatory mediators. So these investigators didn't study it, but it could be that there could be effects of this particular industrial toxicant on contraction or relaxation in the airways. And then more recently, there is environmental toxicant exposure or industrial toxicant exposure. We use this model to study the effects of salicylic acid, which salicylic acid is in like a lot of cosmetics. And then also toluene diisocyanate, which is TDI, which is a toxicant that's released during process of processing of plastics, and found that salicylic acid, but not TDI, actually increased reactivity of the airways to a contractile agonist, which we thought was kind of interesting. And we're examining exactly how that occurs right now. We've also used, and others have used, PCLS in the study of a thing called idiopathic pulmonary fibrosis. So it's basically thickening of the lungs and destruction of the airspace. And what you can see up on the top left corner is actually lungs from an IPF donor that we had received. What you see in the bottom left is if you stimulate with TGF-beta, which is known to be a pro-fibrotic agent, you get an increase in the amount of a number of different fibrotic endpoints, including fibronectin, PI1, collagen 1A1, and that if you use a receptor antagonist for the TGF-beta 1 receptor, the SB compound, you actually decrease it to baseline. And interestingly, although TGF-beta has been primarily known to be a fibrotic agent, we also found that TGF-beta treatment actually increases contractility of the airways and decreases relaxation of the airways. And so what you see here is TGF-beta increases not just only baseline tone, but increases the contractility of the airways to a contractile agonist. And on the flip side of that, if you're looking at relaxation of the airways, both TGF-beta and FGF2, again, a profibrotic, or mainly known as a profibrotic agent, actually decreases relaxation to a beta agonist. So in summary, we and others have used PCLS as an integrated tissue model. We study asthma, COPD, and IPF, but there are a number of other inflammatory diseases or lung diseases that are used with this type of model. We've looked at efficacy of current and emerging therapeutics to treat airways in this system. And then we've also looked at precipitants of lung diseases and looked at airway reactivity, to try to understand how airway hyperresponsiveness and inflammation that's associated with exposure of whether it's environmental exposure, industrial exposure, or other precipitants like viruses of worsening of asthma. How does that affect the airways? So there's so many people that I could acknowledge that it's just kind of unbelievable. But these are just some of the few people that have been very critical in all of the studies that we've done. And I guess with that, I will take questions.
Wonderful. Thank you so much.
Yeah.
So everybody, I don't have any questions or comments in the chat box. If you still want to ask questions, feel free to enter it there. We have a bit of a small group, so right around 20 people. So if you have questions, please do feel free to show yourself on video and unmute yourself and ask. Does anybody have any questions for Dr. Koziol-White?
I have a handful of small questions, just so I know more about this. They kind of range from different things. For one of the endpoints, you put like area under the curve. This is in your like PFK 15. At first, I didn't know what to think of that. Could you tell me a bit more? Is that just kind of like adding up every contraction in the end that you get?
Yeah, so it's sort of the integrated response, and what we do is we calculate it from the sigmoidal dose response curves that we generate from the data. So yeah, so it's sort of the response across the entire dose response to carbachol or to formoterol or something like that.
Okay, so I guess it like covers up for if one slice starts reacting at a little lower dose than the next.
Yeah,
and then you could kind of okay. And then how much time do you need to pre-incubate with PFK 15? Like what's a pre-incubation typically if you want to study bronchoprotection?
Yeah, usually for bronchoprotection, it's somewhere along the lines of 30 to 60 minutes. And in some cases, if we're looking not necessarily at bronchoprotection, but if we're looking at something like enhancement or like the effects of an inflammatory mediator, which would probably be, transcriptional, you need transcriptional induction of different things. Those are usually somewhere between 6 to 24 hours.
And then let's say for the PFK 15, are you washing it out because it normally is cleared? Or
Yeah. So these like the dose response is usually done, most of the time is usually done in like where we're replacing the media and then measuring the constriction or relaxation.
Okay. Yeah, okay, that makes sense. So you clean it out. And then one thing I think about too is like doses of I don't know if it's cytokines or mediators, but you have a lot of cases where you put like TNF alpha, IL-13 or TGF beta. How do you determine what dose you should be putting of those to make sure you're relevant?
Yeah, so it's a lot of it's normally looking through the literature and trying to determine from levels of, or what's I guess an acceptable level or whatever that's been examined in bronchoalveolar lavage fluid from asthmatic patients. So what we try to do is we no matter what inflammatory mediator that we're like working with, we usually will do a dose response and then settle on a single dose and then try to determine whether antagonism of a receptor or a small molecule inhibitor or something like that would actually change the response with. We try not to have like a maximal concentration that you wouldn't be able to inhibit anything with. We try to pick sort of a middle dose. Okay,
so it's VAL guides you. Then you make a dose response and you put the middle.
Yeah, kind of a middle dose. Yeah. So what we'll try to do is we try to titrate it down to almost like a no effect dose, so that we kind of figure out what range we're like is appropriate to work in.
Yeah. Okay.
Yeah.
Great. Hey, Dr. Koziol-White, we do have somebody who wanted to ask a question, which is, could you comment on the use of Precisionary instruments microtome versus other microtomes, such as advantages and disadvantages? And I just wanted to add that ours is called a compressed-tome, which Dr. Koziol-White referred to. And the other main PCLS lung slicer is the Crumdiac. So I don't know, Cynthia, if you could comment on those.
Yeah, actually, we used the crumb deck for quite a while, actually, before I kind of had started postdoc and stuff with Dr. Panettieri, and found that actually this compressed-tome was not only able to produce really nice lung slices, but we were able to produce a lot more with the compressed-tome than we were with the crumb deck. I think mainly because so the crumb deck, I mean, it does have its uses and stuff. It's a nice instrument, but we found that when we inflate the lungs with the low melting temperature agarose, we were only able to actually slice the cores from from a given donor or something like that for about a day. Like that, like during the day after we had inflated the lung and everything and processed the tissue, whereas the compressed-tome has actually been able to afford us the ability to actually make slices for a couple of days. So we've increased like the output from the lungs significantly using this compressed-tome.
Awesome.
Yeah.
Thank you so much.
And actually, we were introduced to the compressed-tome by a collaborator of ours at University of Arkansas for Medical Sciences. His name is Richard Curtin. He actually had been using it for a couple of years before we were sort of turned on to it. And I like, am forever indebted to him that we did make the switch.
Got it.
So,
Wonderful. Wonderful.
yeah.
All right. If there are no more questions, go ahead.
I have a question there. Sorry, I can't put on my video. But the point that you mentioned about being able to use the lungs for a lot longer, could you just tell me a little bit more there in terms of versus being able to use it only for 24 hours?
Yeah. So I think that the reason that we're able to do this more so with the compressed-tome compared to the crumb deck is that the way that the sample is loaded into the crumb deck, it's actually loaded so it's the same diameter as the pedestal that you put it in order to slice it. And so what we were finding is if you tried to slice from cores like the next day after you had inflated the lungs and then processed the tissue and stuff like that, we normally store the cores in a transplant buffer that the lungs come in, just because it's it's an environment where there's a nice pH salt balance solution and stuff like that. It's transplant buffer that it's buffer that transplant organs and stuff like that is transported in anyway. So it's a kind of a better environment than just putting it in like something like PDS. And then is in terms of using the compressed-tome and being able to slice the cores for days afterwards, what we found is that the diameter of the core of tissue that we're generating with using that little cork boring tool, and then the diameter of the specimen tube that we loaded into, the specimen tube is actually larger than the diameter of that tissue core. So what we're able to do is we're able to take the low melting temperature agarose that we inflate with, and we're able to surround the core so it sort of stabilizes it better.
Mm-hmm. So, okay, so you prepare the loading pistol with agarose and then save it in the environment that you mentioned with the transplant buffer.
Well, so what we do is like we inflate the lungs with the agarose and then process the tissue and make the core, like the tissue cores. And then we store the tissue cores actually in the transplant buffer, overnight or something like that, so that we can actually continue to slice the next day. But we try to get through as many of the tissue cores on the first day that we're inflating the tissue and processing the tissue. But we can take those cores, put them in the specimen tubes and surround it with agarose so that we can make sure that it's a little bit more rigid when it comes to slicing it. And it helps keep the integrity of the tissue. And so you get really nice airways still,
Okay.
even a day after. So.
Okay. So the cores you put in the transplant buffer and put them on the fridge like 4 degrees.
Yep.
And when you are just preparing the slices, you do the pistol and loading with agarose, the pistol.
Yeah. Yep. Exactly.
Okay. Yeah, that's very helpful. Thank you.
Mm-hmm.
Dr. Koziol-White, we do have another question asking, what is your source of healthy human lung tissue from? And can you do agarose perfusion with just a small section of the lung lobe rather than a full lung, which is not easy to come by?
Yeah, so we've had, I mean, it's a bit of a challenge if you only have a section of the lung, and then also it becomes a bit problematic to try to inflate a section of the lung with agarose, just because you'll have, if you have a cut section, the agarose will come out of the tissue because there's not pleura that surrounds it, sort of to keep it in. So we found that even with the intact lungs that we get, if there's a nick in them or something, we'll have to use like hemostats to try to keep it closed so that the agarose is not, that we're perfusing the lung with, doesn't come out of the tissue. So it's advantageous to have as much intact lung tissue as possible and be able to, I mean, we inflate through some of the larger airways that are more cartilaginous airways. And in terms of where we get healthy human lungs, there's a couple of different tissue repositories and stuff like that that are like commercially available vendors and stuff. One of them is called NDRI, or National Disease Research Interchange. And then the other one is a thing called the Musculoskeletal Research Institute, or the Institute for the Advancement of Medicine. So it's IIAM. Those are our main sources. They actually work with organ procurement agencies across the United States, and actually also in Puerto Rico, to help us obtain both the non-disease tissue and actually the diseased tissue as well.
Wonderful. We are wrapping up on almost the end of the hour. So I don't want to keep everybody, and I just want to extend a big thank you, Dr. Koziol-White, for your time and your presentation of your research. And thank you everybody for attending. Have a good weekend.
Thanks, guys.